Hello, everyone. This is going to be Chapter 2, Part 2, which I hope is going to be much shorter than the first one. So let me see where we left off. I know that we left off talking about carbohydrates, so I'm going to skip to that slide. I think it was something like slide 44. I'll get there in just a minute. Okay, yeah, we left off talking about carbohydrates. I won't go through this again. Just a reminder, we're talking about our nutrients, the nutrients that make up the body. So carbohydrates, proteins, and lipids are really the subject of this second part. And we did finish up carbohydrates. I'm going to go into lipids now. So lipids are hydrophobic. You should know the term hydrophobic. Last time I mentioned that. It means that a molecule or a substance fears water. So this will be a substance that is not going to exist or function very well in an aqueous environment or a water-based environment. The main types of lipids, these are the classes. You have four main classes of lipid: triglycerides, phospholipids, steroids, and eicosanoids. Our main focus is going to be triglycerides and phospholipids. Particularly phospholipids are really important, but we'll definitely mention triglycerides and a little something about steroids, which are not totally understood, actually. So let's start with triglycerides. So when you think of fats, certain foods that you eat, if you read the labels and looking at fat content, really we're talking about triglycerides. That is the class of fat that we're talking about. So you should know the basic structure of a triglyceride. It's going to consist of 3 fatty acids. That's why it's called triglyceride. It's made of 3 fatty acids. And those 3 fatty acids will be bound chemically to a molecule called glycerol. So that's the basic structure of a triglyceride. What are the functions of triglycerides? Energy storage, insulation, protection. So these are all general functions of fat. When we talk about what are the functions of fat or adipose, this is what we're talking about. We're talking about triglycerides. And so yes, lipid or adipose, which is composed of triglycerides, is going to function in these things. All right, these triglycerides can be broken down for energy if needed. Of course, you remember the sequence in which we break down nutrients for energy. We have first carbohydrates, then protein, then fats. So triglycerides will be the last to be broken down for energy. But this is really, to help if you're in a starvation state, let's say starvation state, and you're not eating anything, you're not eating carbs or protein, then your body will start breaking down these triglycerides for energy. So we're going to store energy in the chemical bonds that exist in these triglycerides. Collectively, our adipocytes, or our cells that make up adipose, are going to serve an insulating role. So yes, fat does play a role in body temperature regulation, because this tissue that is made of triglycerides is going to function to keep organs, for example, warm. They're going to help maintain that 98.6 degrees Fahrenheit that we need for our organs to function properly. And then protection is a role, just simply in a mechanical sense, that adipose serves as a cushion. We do find adipose in certain areas. There's a little bit in your joints, for example, that will help with shock absorption, and in this way can serve a protective role. So triglycerides are of types, and you should definitely know this. I know there will be a question on this, on understanding the difference between saturated fats and unsaturated fats. You hear these terms all the time, and again, these are going to be on your labels when you're looking at food products. You'll see what's the concentration of saturated fats or unsaturated. So you should definitely know the difference between these two. These are important to know. So what is a saturated fat? A saturated fat, it gets this name saturated based on the chemical arrangement of that triglyceride. So what that means is the carbons in this fatty acid are going to be linked via single, that's the key word here, single covalent bonds. So basically what you have is you're going to have a carbon, and this carbon can bind to hydrogens. And so that's what your triglyceride molecule is going to be composed of. It's a long chain of carbons that are bound on all sides by hydrogen. Now, I said bound on all sides. So a carbon has 4 places at which it can be bound to. So for a saturated fatty acid, a carbon is going to be bound on all 4 sides of it by a hydrogen. And this is going to be by way of a single covalent bond. So when we say saturated, we're saying that the carbon is saturated with hydrogens because on all 4 sides of that carbon, a hydrogen will be bound to it. So the carbon is considered saturated. It's saturated with hydrogens. No more hydrogens can bind to it. It has the maximal number of hydrogens bound to it. So that's really the sort of the chemical definition of what a saturated fatty acid is. Unsaturated means that our carbon is not saturated by hydrogens. How this is going to look is that your central carbon is going to be bound by hydrogen atoms, yes, but they're linked by way of double bonds to that carbon. Okay, so what happens is you end up having a linear looking structure. And actually, I'm going to end up making just a short document, sort of with a picture of this for you all that I'll put up on Blackboard outside of this to sort of make that more clear. So if it doesn't make sense on the visual on this, I'll be putting that up there. Now, another thing about these, what's the difference between saturated and unsaturated? Well, you all might know that saturated fatty acids are the ones that are considered the bad fats. These are the ones that are not good for you. Well, not good for you to consume in high amounts, right? If you consume saturated fatty acids in more than like mild to moderate amounts, then you have this situation where you have a carbon, these fatty acids with a carbon that are surrounded on all sides by hydrogens, which means that it's going to be much harder for the body to break down a saturated fatty acid. So if it's harder for the body to break it down, that means that fat is going to be stored. It's going to go into your storage in your adipose. And this is what contributes to obesity right here, or gaining weight and fat, is consumption of saturated fatty acids. Unsaturated, on the other hand, are the healthy ones. These are the fatty acids that are good for you because this carbon, the carbon that makes up these molecules, these fatty acids, are not bound by hydrogens on all sides, which means that these are going to be easier to break down. They're going to be much more likely to be broken down and used as an energy source and not stored for later. Okay, so that's why unsaturated is considered better for you. An example of an unsaturated fat, there are plenty of them, is actually omega-3. You've all heard of it. And this is actually an essential polyunsaturated, meaning it has many areas of unsaturation. And it's considered essential. And that when it's essential, that means you can only get it from your diet. Something that's called essential means your body doesn't make that substance on its own. You have to get it from your diet. So omega-3 is really a good one for you. It is associated with a lot of different benefits, especially in the brain. The brain, the neuron associated with nerve impulse conduction in the brain are going to be composed of this omega-3. So it's really good for the brain, for nerve impulse conduction and stuff like that in the brain. Here is a picture of examples here of some fatty acids. And you don't need to memorize this at all. You don't need to know the names of these specific fatty acids. This is just sort of just get a general of what the layout is here of a fatty acid. All right, first of all, a triglyceride. We're talking about the structure of a triglyceride. So we're going to have one fatty acid, a second fatty acid, and a third fatty acid. So that's 3, triglyceride. Glyceride, because these 3 fatty acids are bound to a glycerol molecule. All right, so this is the structure of a triglyceride. Now it is showing you here at the top an example of a saturated fatty acid. And maybe you can see here what I was talking about. So you have a long chain of carbons, and bound on all sides. The carbons are going to be bound on all sides. On all sides. Okay, whether that be by hydrogens or another carbon, the next carbon next to it. The point is that each carbon is considered saturated. That means it is bound on all 4 sides of it. Notice that each carbon is bound to something on all sides of it. It's considered saturated. So you can kind of think, well, yeah, it is bound on all sides, these carbons, so it makes sense that it would be a little more difficult to break down. Part of the reason is because enzymes that do the breaking down of these fatty acids, the enzymes just angle-wise, that enzyme has a difficult time getting the proper angle to get in there and break all those single bonds. All right, it's sort of like interference, right, with the enzyme. The enzyme has a hard time getting in between there. Now, look here at the bottom, it's showing an example of an unsaturated fatty acid. So notice that some of these carbons here are not bound on all sides. So imagine an enzyme coming along, it's going to be much easier for that enzyme to have access to this area where these double bonds are. And so this is why it's going to be easier for the body to naturally break down these unsaturated fats, and why you will be more likely to use these as energy. Just another example of how this is joined together. We have 3 fatty acids joined together with a glycerol molecule. So that was triglycerides. So the next class of fat, or lipid, however you want to say that, is phospholipids. So phospholipids are really important in physiology. We will be talking about this again in chapter 3 when we talk about the cell membrane. So what is a phospholipid? It's sort of like a triglyceride, but it's just that it's missing that third fatty acid. All right, and it has a little something extra on it. So the phospholipid is made of 2 fatty acids instead of 3. It does have a glycerol molecule like the triglyceride has, but it also has this extra group here. It's a phosphorus containing group. Phosphorus meaning that it's containing phosphate. This is why it's called a phospholipid. And really it's this phosphorus group that you'll see will be so important in physiology. Phospholipids are really unique in terms of being in the class of lipid because they are considered amphipathic. These are not hydrophobic molecules 100%. Okay, they're partly hydrophobic. They're amphipathic, meaning both. Okay, so the phospholipid can exist in both a hydrophobic and a hydrophilic environment. So the molecule is going to consist of a head region and a tail region. The head region is the polar part of the molecule. This is the part that likes water. This is the part of the molecule that will actually reside in a water-based environment. The other end, or the other part of the amphipathic phospholipid, is the tail. And so the tail is actually the nonpolar part. So this is the part that will be repelled by water. This is the part that will exist in a lipid-based environment. And again, the phospholipid and the fact that it's amphipathic and this specific structure of having a polar head and a nonpolar tail are really important in the cell membrane structure. Here's a picture of a phospholipid. So you see two fatty acids, one here and one here. It is bound to a glycerol molecule, but on the other side of that glycerol molecule, we have something extra. We have that phosphate group. So the phosphates, I don't know if you can see on the slide, but there is a phosphate here. Okay, so that's considered your phosphate-containing group. So what about the head and the tail regions? So the tail region is indicated here in this bracket. The tail region is composed of the two fatty acids. So the tail region is considered hydrophobic. This is the part of the phospholipid molecule that is going to exist in a lipid-based environment. This will be repelled by water. The head region is the other end of the molecule, and that is the phosphate-containing group here. So the phosphorus-containing group is considered the head region or the polar region. This is the part of the molecule that is able to exist in water-based environments. And we'll see when we look at cell membrane structure why that arrangement is so critical for the structure of that plasma membrane. So a little bit about eicosanoids. Not going to dwell on this too much because it's a lot really isn't known about it. Eicosanoids, though, are derived from something called arachidonic acid, which is found in your cell membranes. Again, we're still trying to understand how these eicosanoids work. It's a relatively new class of lipid, but it has been found that they do hormone-like signaling. So again, we're still trying to elucidate what's really going on with the eicosanoids, but they do have some sort of hormone-like behavior. Prostaglandins are a kind of eicosanoids. We do know a little bit more about prostaglandins. Prostaglandins, again, these are going to originate from the cell membrane. Prostaglandins play a role in healing. So they play a role in inflammation, they play a role in blood clotting, and some other issues. Okay, and that's really all we're going to say about eicosanoids. You should know that prostaglandins are a kind of eicosanoid, and prostaglandins are, because we know a little bit more about them, are often a target for drug action when we're dealing with things like inflammation, for example. Steroids. Steroid is your fourth class of lipid. So steroids exist in a ring-like structure. Whenever you see like a big ring structure, and it's a lipid, it's going to be a steroid. The most important kind of steroid for us to understand first is cholesterol. Cholesterol, you've all heard of it. It is actually, it says here, the parent steroid from which all other steroids are synthesized. So in the body, when you're making other steroids, such as steroid hormones like testosterone and estrogen and similar steroid hormones, the basis for the synthesis of that is cholesterol. So in other words, you have cholesterol in order to make other steroid hormones in the body. All steroid hormones are going to be formed first from the cholesterol molecule. So what that tells you is, well, you need cholesterol. Everybody talks about how bad cholesterol consumption is, and it is bad because here's the deal, and a lot of people don't know this. We make our own cholesterol. Cholesterol is not considered an essential substance, meaning we make it already. So you don't need to consume any cholesterol because you already make it. You already make it using triglycerides. Triglycerides and other molecules are going to come together and make cholesterol. So you make it all by yourself, which is why it is bad to consume a lot of cholesterol, because what will happen is that cholesterol is not going to be needed by the body because you already make enough. All right. As it says here, 85% is internally synthesized. That's most of it. All right. about 15% or so comes from the diet, and you definitely don't need any more than that 15% because 85% takes care of it. So again, what happens when we consume extra cholesterol from our food is that extra cholesterol builds up in our arteries. It's going to build up in our arteries and other locations. And this is what ends up over the long term causing very bad problems like atherosclerosis. You've maybe heard of atherosclerosis, which is referring to a hardening of the arteries because we get plaque formation. Cholesterol builds up in the arteries and forms plaques, and those plaques don't break down. All right, they just stay there in the arteries and block blood flow. So atherosclerosis in the long run can actually lead to things like heart attacks because those plaques are blocking blood flow. So you really can get a lot of this cholesterol buildup around the arteries that supply the heart. So if you're preventing blood flow to the heart, this can lead to heart attack or other types of heart disease. So this is why, you really don't want to have a lot of cholesterol in your diet. It's bad. Other steroids, vitamin D. We've talked about vitamin D, and vitamin D is definitely a steroid. Other ones that you've probably heard of, cortisol, progesterone, estrogens, testosterone, bile acids. Bile acids are going to help break down or emulsify fats in your diet. But just be familiar, we're not going to go into detail about any of these. We did talk a little bit about vitamin D, but you should just know these are examples of other steroids that we have in the body. Here is a picture of a steroid, all right? Again, it's going to have like this ring structure to it. Now it says here, okay, so this is more has more of a clinical application for you all. All right, it's okay. There's only one kind of cholesterol. You have people out there saying this thing called you have good cholesterol and you have bad cholesterol. It's sort of been categorized, but understand that that isn't two different kinds of cholesterol. All right, there's only one kind of cholesterol. Okay, and again, in the body, cholesterol does more good than harm because we make all of our steroid hormones and those kind of things. Okay, so in that sense, cholesterol does more good than harm as long as you're not consuming a lot in your diet. In the body, all by itself, cholesterol does good things. Okay, so what do we mean when we say like good cholesterol and bad cholesterol? We're not talking about the cholesterol itself. We're talking about something called lipoprotein. Okay, Lipoprotein is something that is made of more than just cholesterol. It's made of cholesterol, fat, phospholipids, and proteins, hence the name lipoprotein. So what are these lipoproteins? Lipoproteins actually attach to and transport and store cholesterol. Okay, so basically consider these lipoproteins like they transport cholesterol around the body. All right. So this is where we get into our good versus bad. So what's the difference? Good is what you would want to know as HDL, or what that stands for is high density lipoprotein. This is considered the good cholesterol. And the reason it's considered the quote unquote good cholesterol is because this particular lipoprotein has less lipid in it. It has more protein and less lipid. All right, so that means it's gonna float around the body easier. It's not gonna get stuck in your arteries and stuff like that. If it has a lower fat concentration in that lipoprotein compared to protein, then it's going to just move around the body faster. And again, what are these lipoproteins? They're helping to transport things like cholesterol. So HDL is better for you. All right, it has been linked to lower incidences of cardiovascular disease, and because we're able to use this transport mechanism, lipoprotein, with the lower fat ratio compared to protein, it's just going to be easier for this transport protein to flow around through the body. It's not more likely to get stuck because lipid's heavy, right? If it has more lipid, such as LDL, if it has more lipid, it's slower. It's more sluggish to move around in the bloodstream. So it's more likely to get stuck somewhere, all right? So low density lipoprotein is considered your quote unquote bad cholesterol, even though it's not cholesterol. Remember, this is a lipoprotein that is partly made of cholesterol and helps transport cholesterol, but it's not cholesterol itself. So this transport protein, lipoprotein LDL, if it's a low density lipoprotein, this means there is a lower concentration of lipid compared to protein. Low density lipid. All right, or you can say it high ratio of lipid to protein, meaning there's more protein. Okay, so this is a little bit easy. This is a little bit. I actually said that opposite, right? It's a low density lipo. Let me rephrase this so that it's easier to understand. When you talk about high density versus low density, you're referring to the concentration of protein. And I think I said it backwards previously. If I did, I apologize. I'm having a kind of a quick day here. So high density means we have more protein. And again, everything I said still applies, to this. The fact that this is considered the good one, right? Because you have more protein compared to lipid. So the word high density is referring to how much protein is in it. So I hope that makes a little more sense. So low density lipoprotein means that we have a lower density of protein compared to lipid. So that's why these are going to be, may contribute to cardiovascular disease because they're going to be slower to move around. So not a big deal on these. You should just kind of, know the basic difference between these. So let's move on to proteins. Protein is a polymer made of individual subunits called amino acids. So amino acids are going to come together and build these larger proteins. Proteins are very important in physiology because they have the most varied functions of any other molecules, and we'll be mentioning many different types of proteins throughout the course. Proteins can have many different roles. They can play structural roles, chemical roles. For example, enzymes are classified as proteins, and enzymes are going to, as you might recall, speed up the rate of chemical reactions. Contraction, there are all kinds of proteins that are required for muscle contraction. So the polymers of an amino acid, which basically is a protein, so amino acids are monomers. And these monomers are going to be chemically bound and held together by a specific type of chemical bond called a peptide bond. So amino acids join together by way of peptide bonds to build up a larger polymer or protein. So proteins are going to have different shapes and different functions, and a lot of this has to do with different levels of structure that amino acids have, all right, which we'll go through the 4 levels of structural development, basically, of a protein. All proteins are made from a combination of 20 different types of amino acids. You don't need to know all those amino acids unless you take biochemistry. You will need to know all of those and the structure of them. But here, just know there are 20 of them. But you definitely want to know that each amino acid contains these special groups. Each amino acid contains an amine group, and it contains an acidic group, also called a carboxyl, and we'll see that on the next slide. Okay, so again, the monomers or the amino acids are joined together by peptide bonds. Now, the peptide bonds are going to form based on a joining of the carboxyl group of one amino acid with the amine group of the next amino acid in the sequence. Of course, we're building something up, so this is going to be done through the process of dehydration synthesis. This is an anabolic reaction which will require energy, so it will be considered an endergonic reaction. So let's look at a picture of this. Really important to know. Definitely want to know this structure. I'm sure there'll be a question somehow about this. So this is the basic structure of an amino acid. Remember, the amino acids are the monomers or the building blocks of the larger proteins. So you're going to have a centrally located carbon. To the left of that centrally located carbon, you will find your amine group. The amine group is going to be nitrogenous, meaning it contains a nitrogen. And so it's basically written out as NH2. So you'd have one nitrogen and two hydrogens, NH2. On the other side of that centrally located carbon, you have your other group. This is the acidic group, also called the carboxyl group. So here we're going to have carbon double bonded with an oxygen and then single bonded with an OH. Now the OH together, OH you just commonly call a carboxyl. That's why we can also call this a carboxyl group, right? The R out here is meaning there's going to be some other entity out here bound to this amino acid, which gives this amino acid its identity, its specific identity, right? Because remember, we have 20 different amino acids. So it's this R group that will give it its identity, whether it's valine or cytosine, whatever. But here is the general universal structure of the amino acid. So what's going to happen when the amino acid joins with another amino acid during dehydration synthesis is that the acidic group of this amino acid, for example, is going to bond together with a amine group of the next amino acid, which is not pictured here. And that would be called a peptide bond. And here's a picture of a peptide bond being formed. So here's one amino acid. Notice we have our carboxyl group here. We have our second amino acid over here, and it has its amine group on the other side of it. So what will happen is that this carboxyl group of amino acid 1 will bond with the amine group of amino acid 2. How do we do this? We do this through dehydration synthesis. We're actually going to remove water, and that's what's shaded here in blue. We're going to take a hydrogen off of amino acid 2 and a carboxyl group off of amino acid 1 equal H2O, or water. We remove that so that we don't have an obstacle anymore between these two amino acids. Now they can come together and bond. And once that takes place, all right, we can say we have formed a peptide bond. Here's just another way of looking at the same thing. You've got two amino acids here. We're going to use dehydration synthesis, the removal of water. We're going to form a peptide bond after that. The peptide bond is between carboxyl group of the first amino acid in the sequence and then in the amine group of the next amino acid in the sequence. Now, if this is reversible, which, yeah, we can break our peptides, we can break our proteins down into individual amino acids like we do during digestion. And so this would be done through hydrolysis. We can break this dipeptide down into its individual amino acids by adding water. We're going to add water, create a disruption here in this peptide bond. That water will separate these two, and then you will have them as individual amino acids. Couple of points about proteins in general. Conformation. So conformation refers to the three-dimensional shape of a protein. And it really is this conformation that makes a large contribution to the function that that particular protein will have. Now, proteins will often change their conformation. And most of the time when that conformation is changed, that's how it's going to perform some sort of function. We are going to see how this happens several times here in this class. So basically what will happen is that we'll have a protein and something else is going to come along and bind to that protein. And anytime something else comes along and binds to a protein, okay, another chemical, another ion, any kind of substance that binds to the protein, it causes that protein to change its shape. It changes its conformation. And then that sort of activates the protein, and that will cause it to perform some sort of function. And again, we'll see that. We'll see many examples in the future when we actually go over those. Denaturation is just another concept relating to protein. Basically, protein can be destroyed, or the conformation of the protein can be destroyed, the shape of it. And most often this can be due to extreme heat or extremes in pH. So the structural levels of proteins, got to know these for sure, will be a question. There are 4 different levels of protein structure. Okay, so basically this is referring to the development of a protein. The protein will go through 4 different steps of existence. The first level of existence, or the first level of protein structure, is referring to the linear sequence of individual amino acids, or the order of the amino acids in a long linear chain. Next, we have the secondary level. So what ends up happening is that this linear sequence of individual amino acids, those amino acids start to interact together. And so we start to see the development of more bonds, all right? And we'll start to get coiled shapes, all right? The linear sequence will start to sort of bend over on itself. And this is due to the formation of hydrogen bonds between the different amino acids. You should know there are 2 types of secondary structure. You have alpha helix and you have beta pleated sheets. Alpha helix, and we'll just look at a picture of them. I'm not going to ask you to recognize what these look like. You should just know that alpha helix and beta pleated sheets are types of secondary structure. We'll see a picture of it in a minute. The third level, so once we have this secondary structure formed, then we will undergo our tertiary level of development. And this is when our secondary structures start to interact with each other. So the beta pleated sheets and the alpha helices start to interact with each other, the formation of hydrogen bonds and other bonds. Now, the tertiary level of development of a protein is very clinically important. And I do want to mention here, so we're going to have another classification of proteins. We will classify proteins into globular proteins and fibrous proteins. And it's really at this level when you can start to make this classification. So, but clinically, why is the tertiary level important? And the reason for that is because a lot of errors, a lot of mistakes can happen during the development of a protein at the tertiary level. Basically, having a lot of protein folding going on. And so, you can imagine there can become a lot of complexity here. Once the alpha helix and beta pleated sheets start to fold around on each other, it can start to get complicated, and there can be errors. And so there are a good number of diseases out there that are directly caused by a problem at the tertiary level of protein development. The protein does not develop properly. You end up with a protein that's useless. It doesn't work. Example of that would be Alzheimer's disease. One of the causes of Alzheimer's disease is an error in protein folding, an error here at this stage. The final level of protein structure is going to be quaternary, and this is the final version of a protein. In a lot of cases, some proteins you don't have that quaternary level. Most proteins, in fact, will stay at the tertiary level, but other proteins will advance to an even more complicated structure. And this an example of that, which you'll study in 139, is hemoglobin. These are pictures of each levels. So this one here is showing you the primary level of organization or development of a protein, and it's just the individual amino acids in a linear sequence. Then you have your secondary level where we start to form. Now here is an alpha, okay? and that one looks like a spiral. It's like a coil. Then you have your beta pleated sheet, and it's just like it will look like it's forming like this sheet arrangement. Then you have the tertiary level, which again is the really important one, and then you have your quaternary, and this one is showing actually the structure of hemoglobin. Here's just another way of looking at it, okay? Primary sequence, individual amino acids. Secondary structure, we're having the formation of an alpha helix, or you may have the building up of a beta pleated sheet, okay? And again, this is just due to vein interactions between all the amino acids. And then tertiary, so tertiary, again, clinically important. This, in a lot of cases, is going to be the final version of a protein, representing how the secondary structures interacted with each other, and here you have it. And then you may actually get an even more advanced structure to the protein, and that would be the quaternary structure. So I mentioned that at the tertiary level, we can start to classify proteins into two classes, fibrous and globular. So definitely going to be a question on this, really important, need to know the difference between these. So the class of fibrous proteins are considered structural proteins. That means they don't really function, they don't have a physiology about them. They really just serve a structural role, fibrous. So these are typically going to be water insoluble. That means they are going to not be, well, they're going to be hydrophobic, right? So what's the purpose of these? They're going to provide mechanical support to tissues. They're going to provide strength to tissues. Definitely want to know the examples of fibrous proteins that I've put down here. We will be talking about all of these. So I'm not going to go into detail about them now, but you should know these are examples of fibrous or structural proteins. You have keratin. Keratin is a very hard, tough protein that you find in the skin. Elastin. Elastin is also a structural protein that provides an elastic sort of structure to a tissue. Elastic. It has elasticity. And then there's the very important collagen. Collagen is the most abundant protein in the whole entire body. All right, but understand it strictly serves a structural role. It is a fibrous protein. So the second class is the globular proteins, and these are the functional proteins. All right, these proteins have important functions. They are going to be water soluble. The reason for that, okay, so they're going to be hydrophilic. Reason for that is because they have to be able to be transported through the bloodstream. Globular proteins have to be able to be transported from one site to another, whereas fibrous proteins just stay where they are. That's why they don't need water soluble, because they don't need to go through the bloodstream. They're going to stay right where they are, contributing to the structure of a tissue or an organ. Globular, on the other hand, are going to be water soluble. All right, these examples you should know, and we'll talk about specific ones throughout the course. Antibodies, we need to get antibodies from place to place. So these are proteins, and they're globular proteins. Hormones are proteins. Molecular chaperones. So molecular chaperones are proteins that transport other substances throughout the bloodstream that are not hydrophilic. So let's say, for example, you have a protein or a lipid or something that is hydrophilic. It can't go through the bloodstream because it's not capable of being in a water-based environment such as the bloodstream. In that case, that substance will be carried through the bloodstream by a molecular chaperone. So chaperones are really cool, and yes, they are globular proteins. Enzymes are also globular proteins. So, again, we're some detail here about some of these, like keratin and collagen I mentioned. Don't worry so much about where you find these right now, okay? Just be concerned that keratin and collagen are examples of fibrous proteins. We'll get into the details of those later. Again, hormones I mentioned are globular proteins, and they're associated with communication. Hormones are going to help provide signals between cells. Receptors. So receptors on the surface of cells play a role in communication. All right, and receptors are proteins, or they might be partly proteins, okay, but they play a role in communication. Membrane transport, we will talk about this in chapter 3, so don't worry so much about understanding these right now. But in general, in your membrane, your plasma membrane around your cell, you have these carriers, or you have these channels that they are proteins, and they're going to help transport substances across the cell. Enzymes, like I mentioned, are globular proteins. Again, just going through some of the important functions of proteins. So yes, recognition and protection. Antibodies are a type of protein, all right, and they're going to be associated with protecting the body as some called glycoproteins. Again, we'll mention this later in chapter 3, so don't worry about it so much right now. A glycoprotein is going to help in recognition. Again, we'll get into that later. Movement. So you have proteins called motor proteins that especially we'll see with the with muscle. All right, don't worry about it too much. Okay, but it's motor proteins are a type of protein involved in movement. Cell adhesion. We have certain types of proteins that are going to be responsible for holding cells together. All right, so just as you can see, there are a lot of different functions of proteins. Enzymes. So enzymes, globular proteins, we already mentioned before that these are catalysts, which means they increase the speed of chemical reactions. So something, a definition you should have associated with enzymes, and it's this. So enzymes are going to lower what I call, and what a lot of people call, energy of activation. Okay, so here's the deal. When you're doing a chemical reaction, when you're starting, you're trying to initiate a chemical reaction, a certain amount of energy is required for that. Well, enzymes lower the amount of energy required for that. So that's why they're really special. Enzymes lower the amount of energy that's required to activate a chemical reaction. So it's in this way that they increase the speed of a chemical reaction. All right, this is going to allow for a lot of chemical reactions to occur. So some other aspects surrounding enzymes. Some enzymes, in order to function, may require a cofactor or a coenzyme. And just in general, you should know these are going to be things like vitamins, or it might be an ion, okay, that will need to be present in order for that enzyme to function. Enzymes are very specific on what kind of reactions they initiate and what kind of substrates they work on. You should know how enzymes are named. So most enzymes are going to end with this ASE at the end of it. And also the name of the enzyme indicates what kind of a reaction that enzyme catalyzes. So hydrolase, right? Hydrolase means that this enzyme, hydrolase, is going to initiate or speed up a chemical reaction associated with water, such as taking water away or adding water. Same with oxidase. We're dealing with an enzyme that works on adding or removing oxygen to a chemical reaction. Here's a picture of this, what I was referring to as activation energy. All right, so on the left, you're seeing a situation, a chemical reaction trying to occur with the presence of an enzyme, no enzyme. Okay, so we're going to start here. It says free energy to start this reaction. Okay, we're going to need even more energy here. Okay, we're going to need even more energy, and it's going to take longer for that chemical reaction to proceed because there's no enzyme. All right, now just compare that overall with what you're seeing on the right. What you're seeing on the right is a situation where you have an enzyme, and this line up here doesn't go up so high. All right, because our activation energy is lower. Our activation energy is less on the right compared to the left. So enzymes lower or decrease activation energy. Activation energy is simply referring to the amount of energy needed to activate a chemical reaction. So enzyme action, again, I mentioned enzymes lower activation energy. So there are three main steps involved in enzymes action. So first we're going to have a substrate, okay, which the substrate is whatever the enzyme is working on. The substrate is going to bind to the enzyme at a specific place on the enzyme called an active site. So the substrate binds to the active site. Once this happens, we then refer to this as an enzyme-substrate complex. At that point, the enzyme is going to do something to that substrate. It might break it down, it might digest it, it might rearrange it, it might be adding substrates together, whatever the case. The enzyme is going to do something to that substrate and then release a different product. I can't really even see this slide, but what this is basically giving you, okay, here's an example of the steps of an enzyme. And the example is sucrose hydrolysis. Right, so we're going to break down sucrose. Sucrose goes to the sucrase. Sucrase is the enzyme for this. Sucrose is the substrate. Sucrose goes to sucrase's active site. All right, and molecules bind together. Okay, we're going to form an enzyme substrate complex. Then the enzyme is going to break bonds within that sucrose molecule. And then at the end, you have a chain. You have the individual units of that sucrose. I'm not going to ask you the details about sucrose on that. Here's a picture, basically what you're seeing. Here's an enzyme. Here's the active sites here. Here's the substrate we're working on. In this case, it looks like we're going to break this down. Once the substrate bound to the enzyme, we call it an enzyme substrate complex. The enzyme will work on the bond, like break it down, and then we have different products released. Acids. Okay, so I forgot to mention nucleic acids as a major nutrient. So nucleic acids, going to try to go through this somewhat quickly. Nucleic acids are composed of nucleotides. These are the monomers of. Nucleotides are going to be made of these things. You should know what the nucleotide is made of. It's going to be made of a nitrogenous base or a group that contains nitrogen. A pentose sugar. Pentose means 5. So it's going to be a carbohydrate that is in that's in a ring that has 5 carbons. 5 carbon carbohydrate and a phosphate. ATP is the best known nucleotide. DNA and RNA, okay, these are our main classes of nucleic acids. So DNA we need to know a little bit about here. So what DNA is basically the genetic blueprint. For the synthesis of proteins, DNA directs the synthesis of proteins. You should know the basic characteristics of DNA. First of all, it's double-stranded. You should also know that DNA is stored in the nucleus of the cell. You should know that DNA contains, okay, so its carbohydrate is called deoxyribose. That's the type of pentose that is in DNA. It's called deoxyribose. You have your phosphate group, and you're going to have one of 4 nitrogenous bases. You should know that the purines and the pyrimidines, okay, these are classes of nitrogenous bases. So the purines, there are 2 types. You have adenine and guanine. Pyrimidines, you have 2 types. You have cytosine and thymine. All right, so DNA, right, is going to have all these things. It's going to have one of the 4. So DNA will have one of these 4. And so what happens to actually form the double strand? Remember, it's a double-stranded molecule. So one strand that has one purine on it is going to bond together with a pyrimidine on an opposing strand, form a double helix. And you should definitely know, I'm sure you'll have a question, about what's known as complementary base pairing. So these bases, purines, are going to bond with pyrimidines. And it goes like this. Adenine is a purine. It always pairs with thymine, a pyrimidine, on the opposing DNA strand to form a double strand. Guanine is a purine that's going to pair with cytosine. And I'm trying to move to the next slide. There we go. So this is showing you the double helix, all right, the double-stranded helix of the DNA molecule, the purple long strand. Okay, you can see that there are two of those, all right? And on each strand, there is a purine or a pyrimidine, and those are going to bond together, all right? G with C, A with T, always. And that's what forms this double-stranded molecule. You don't have to worry about all the chemical details up there. What about RNA? RNA carries out the instruction of DNA, all right? So RNA is outside of the nucleus, right? DNA is inside the nucleus. But we need the genetic information from DNA to go outside of the nucleus so that we can go outside to form all these different proteins we need to make. So it's RNA that serves as the messenger of the DNA genetic code. RNA is going to translate the information from DNA and bring that information outside of the nucleus because it can go outside. Okay, so RNA is a single-stranded molecule. So be sure to know the differences between DNA and RNA. DNA is double-stranded, RNA is single. DNA is inside the nucleus, RNA is outside. And then as far as the makeup here. So RNA, instead of deoxyribose for its carbohydrate, contains ribose. And now we have something different here. So thymine, we do not find thiamine in RNA. We find uracil instead. So what that means is if an RNA is trying to read a DNA molecule, the uracil, okay, is going to, the uracil in RNA is going to read the thymine on DNA. Now there are different kinds of RNA. We're not going to go through the different types. Just know in general that your RNA is basically a messenger. It's gonna translate the message that DNA has and transfer that information out to the outside of the nucleus. Okay, the last thing I have here is really important, and it's about ATP. So I mentioned before, ATP is the cellular source energy, all right, for the cell. It's very critical in order for us to perform all kinds of physiological processes. Okay, and here this is really important. So how is it used as an energy source? ATP, like any other molecule, holds energy in its covalent bonds. It holds energy in there. Okay, so what's going to happen is, first of all, ATP, what does it stand for? It stands for adenosine triphosphate. Triphosphate, that means it has 3 phosphates on it. And what's going to happen is that the second and the third phosphate group are going to be separated from each other. And so when we break the bond between the second and the third phosphate, as with any time we break a chemical bond, energy is released. Because remember, energy is stored inside of chemical bonds. Okay, so we break off the terminal phosphate or the third phosphate from ATP, and energy is released. And it's released extremely quickly, and that's what we're going to do with ATP. And so this is kind of a repeat of what I said, right? Chemical energy is stored in chemical bonds. ATP is no exception to that, okay? So what we're going to do is we're going to break the bond between the second and the third, or the terminal phosphate, from that ATP. All right. Now, energy will be released due to the breaking of that bond. But also, what happens to that terminal phosphate? What happens to that third phosphate? That third phosphate can be transferred and attached to other compounds to activate those compounds. So it's another way that ATP can cause action to happen. Very important that you know this. When we have this loss of a phosphate group, we are then essentially converting ATP into ADP. ADP is adenosine diphosphate, di for two. Okay, so when you lose the phosphate, we convert triphosphate to diphosphate. Now, ADP, what about when we have ADP? Now, once our physiological process is over with, once we've achieved whatever goal we need to achieve in the cell, we add that phosphate right back to it to reform ATP. Okay, so we're at the end of this. So it's a cycle. ATP will lose a phosphate, then it will gain a phosphate, and so on, right? So addition of a phosphate group will convert ADP back to ATP. So this brings us to these two important terms on this slide. So what's the chemical process here with breaking this phosphate? Well, it's hydrolysis. Okay, we're bringing water into the situation. We're taking water and we're adding it to ATP to break off that terminal phosphate. This is done by an enzyme called an ATPase. We're going to break that third phosphate off of there, and we end up with ADP plus that free phosphate plus that energy that came from the breaking of that chemical bond. I want to add to this how important it is to drink water. Everybody's always talking about how important water is for you. This is one of the reasons why. If you do not have water in your body, you are not able to perform hydrolysis of ATP, and that's going to cause you to be very low on energy, right? So people will notice if they drink, their 6 glasses or whatever of water a day, you just feel a little bit more energetic. It's not like what a Red Bull does, but you still have just overall more energy. When you don't have enough water, you're not able to perform ATP hydrolysis. Therefore, you're not able to get the energy that is derived from the breaking of that terminal phosphate off of there. Phosphorylation and dephosphorylation. So just in general, phosphorylation means you're adding that free phosphate, that phosphate that was removed from ATP, you're adding that free phosphate to another group of molecules. All right, adding a phosphate to something means phosphorylating it. And you should know that the enzyme that carries this out is called a kinase. And kinases are very important in physiology. If many of you go into pharmacology and study drugs, you're going to be talking about kinases all the time and phosphatases all the time, right? So dephosphorylation is the other way. Dephosphorylation is when you're removing that free phosphate group from a molecule. And this is done by enzymes phosphatases. Here's a picture of the ATP molecule. It's composed of adenine, which I don't know if that sounds familiar, but it's actually part of it's a nucleic acid. So adenine is attached to ribose, and then we have our ever important triphosphate out here. Look at all the phosphates. We have 3 phosphate groups. So here's another look at this. We have our adenine group, we have our ribose, and really the most important part of the ATP molecule for our purposes are these phosphate groups, the triphosphate part, right? So we have 1, 2, 3 phosphates. Here, between that second and that third phosphate, between the second and the terminal phosphate, we're going to break the bond between them. And this phosphate, the third one, is going to go out, and that's considered a free phosphate. And it will be out there doing other things. And then the energy released from the breaking of that chemical bond. So now we're able to use that energy to do work in the cell. And also that free phosphate that goes out here might be added to another substance, which activates that substance to perform a certain step in a physiological process. This is the last slide, and it's important. You should know this equation. All right, we start with ATP. We're going to add water. Remember the importance of consuming enough water, because you have to have this happen to get, you have to have water to add to this. You're going to do hydrolysis, which is going to result in the breaking off of the terminal phosphate from that ATP, converting it into adenosine diphosphate plus free phosphate energy. Now, this is actually a very reversible reaction. So it's a cycle, all right? This is just going to continually occur. ADP will be reconverted back into ATP. So ADP, we're going to add that free phosphate back to adenosine diphosphate. And to do that, remember, we're going to do basically dehydration synthesis. We need to take that water back out so that that phosphate will fit together with the ADP molecule to reform ADP. Okay, that was it. So I'm hoping it was a little bit shorter than the other one. I'm not sure. But that is it for Chapter 2. I know Chapter 2 is a real beast. It's a lot of stuff. It's a long chapter. But we have finished that as far as lecture goes. So please brush up on all that stuff from Part 1 and 2. And I'll have your quiz for Chapter 2 posted up as soon as I can. Hopefully tonight. I don't know if it'll be tonight, but I'll try. So, all right. Have a great day. I'll see you all later.